All right. I want to be able to use the microphone without having to talk very loudly because my throat is a bit sensitive. Can you people in the back hear me okay? Okay? Can you people in the back hear me okay? Huh? Okay, great. I know that for some of you, you probably have a class that's a long, long way away in a distant galaxy and all that. And that it may not be possible to get here at 3:30 every time. I will usually be starting at 3:30. Today we had a lot of kerfuffle. I would appreciate it if that upper quadrant or upper patch there could be kept clear for people who have to enter late. And then you can come in the back door and you don't need to worry about, drawing attention and stuff. We'll try to have some handouts up there maybe even for that. Welcome. Welcome to 99110, the Introduction to Biochemistry. Biochemistry is a very beautiful subject. It will allow you to integrate the chemistry and the biology that you've learned along the way. It is a subject that is of intimate interest to you at every moment of your waking hours and your sleeping ones too. We are very fortunate this year to have a number of people who can help you learn biochemistry. I'm the course director. I'm Alice Fulton, a professor in the Department of Biochemistry. This is my 25th year at the University of Iowa, and I have a daughter who's graduated, so I've been around the track a bit and picked up a little here and there. We're very fortunate to have several people to help you learn besides me, and I'm going to ask them to stand up as we go along. Our official TA is Susan O'Donnell, and Susan O'Donnell is a graduate student in the Department of Biochemistry, a fairly senior graduate student who also has prior experience teaching biochemical-related subjects. So she is a wonderful resource, and we very much hope you'll take advantage of that. Susan will be available on a recurring basis on Tuesdays and Thursdays, 2:30 to 3:30. This is in a different contrast to the syllabus, and I will include that information in the next handout. The next handout I can change. I think they've already printed the Friday one. 2:30 to 3:30, Tuesday, Thursday, in the head's conference room on the 4th floor of Bullen in the biochemistry department. But biochemistry is so alluring that we have 2 volunteers helping out as well. And I'd like to introduce Carissa Carlson. Carissa was our TA last year. She's interested in long-term being a professor at a liberal arts college, and so she's very committed to teaching and is very enthusiastic and good at that. Carissa will be available on a one-on-one basis. If you want to get in touch with her, give her an email, and you can set up whatever the 2 of you need to set up. We also, for the 2nd year running, have an undergraduate volunteer. Angie, could you stand up so we can meet you out? Here's Angie Hansen. This is the 2nd year that a student has loved the course so much that they've offered to help teach it, and I'm just amazed. So thank you, Angie. Angie has set up a time slot in again on the 4th floor of Bullen in room 333. Her sessions will be fairly structured sessions. I'm not quite sure yet how Susan's going to handle hers. They may have some structure, but they'll certainly be available to answering your questions. And Angie's sessions will start February 1st. So you have many resources. All 3 of these people would be happy to answer questions in other contexts as well. I'm available. So we hope that all of you feel that there are people you can come to if you want to understand something that's not making sense, or if you have big questions. I've raised something in the lecture, and you've got more to ask, and that's great too. So the purpose of this class is to give you a good general sense of the whole discipline of biochemistry. I like to say that biochemistry is 3/4 chemistry, but the biology comes first. So it's trying to understand life at the chemical level. We don't do anything with those interesting green yard reagents. We don't get up into ecology. We focus at this particular level of organization. I do assume that you've had at least a year of college-level biology, preferably the main biology class. And I assume you've had a year of general chemistry and hopefully a semester or 2 of organic chemistry. If you've had those, they will definitely be good resources. If you haven't had them, you can continue to take the class, but be prepared. There may be some things in it that will be more challenging for you than other students. We have a textbook this year in a new format, and I've never seen a textbook done this way. It's probably been happened before. But if you want to buy it this is the principal way it's available through the bookstore, as a 3-hole punch. And so you don't have to schlep the whole book with you every time. You don't have to choose between backbreaking and information. So you can just take out a couple of chapters at a time and travel with it. It's also less expensive than the hardbound version, so I hope you'll find that useful. This textbook was picked because it is designed for a 1-semester biochemistry class. There are a number of biochemistry textbooks out there, almost all of which assume you have a full year to learn it. And one semester classes like this tend to end by throwing out half to 2/3 of the textbook. And I thought it would make more sense to just try to find one that covered what we wanted to cover to begin with. The textbook has in it a number of resources. There are obviously the text materials itself. A number of students have found the review questions that are offered in here. Every general section will have a gold and blue review learning objectives set of questions, and those review questions are very helpful as sort of general questions to get a hold of things. There are a lot of problems to work. Some people learn things better by processing them through problems. They've got that. And they also have something quite interesting, which is a disc. And this disc contains on it some wonderful resources for especially the structural parts of biochemistry. Three-dimensional models that you can manipulate that really make things much clearer than trying to pick them up from just flat, two-dimensional static images. Any questions so far? In addition to this textbook, I will be giving, there are references in the syllabus to online textbooks, and I'll try to be putting together another set of references to electronic resources. A number of people find it helpful to have more than one textbook so that if you don't understand this textbook, you read the same subject over here, and suddenly they both make more sense. So I'm going to be including some electronic versions that you can refer to if you want to do that. This course will have 4-hour exams, 1-hour exams. Those exams are held at the same time as the lecture, so you don't need to try to plan around evenings. There'll be a 1-hour final, comprehensive final, covering material from the whole course. If you need to have a makeup exams are always oral. Trust me, it's hard enough to write one good multiple choice test. Writing 2 good multiple choice tests is beyond my comprehension. And oral makeups are actually faster and easier than written tests. The students who've taken my oral makeups, their makeup scores correlate very well with their written test scores. So my oral makeups, in fact, are testing pretty much the same things. This is obviously too large a class to have formal written assignments that I expect you to hand in any case, it's an upper division class. But one way to give you an incentive for paying some attention to those questions is we will try to use some questions from the textbook on the tests themselves. So, all right, let's see if we can get this to go down. All right, okay. I'd like to offer some suggestions on how to learn biochemistry, and this is based, as I said, on many years of experience in teaching it and learning it myself. Some of it is drawn from psychological research that has to do with any topic. Extensive studies have shown that breaking things up into blocks works better. That if you are going to spend 6 hours learning biochemistry, doing an hour on one day, an hour on this day, an hour on that day, will be more effective than doing 6 hours Saturday night. That is, you can't get the same effect out of putting all the hours together. It would be lovely if it were true. It doesn't work that way. Another thing that can help is to mix subjects together. So rather than trying to study biochemistry for 2 hours, do half an hour of biochemistry, half an hour of history, half an hour of biochemistry, half an hour of history. The switching back and forth between the subjects seems to stir up the neurons and make it easier to fix things in your mind. Interestingly, highlighting and rereading does almost nothing for you. And you may have already had this as a fixed habit for you, but it turns out to do very little. You may feel like you've learned more, but objective tests show that people who use that as their principal study thing have learned maybe 10 or 15% more than they did the first time. So if those don't help you learn, what can help you learn? Well, going back over the material and organizing it for yourself. Instead of simply listening here in class passively, when you take your notes, take them home, reorganize them. Actively trying to connect everything you're learning to things that you already know, so it becomes part of a larger network. The other thing that emerged from this research is something that is one of the sort of sobering truths of life. The methods that are best for learning are not necessarily the ones that feel the best. So you may like doing all your studying for 6 hours on Saturday night, but it is probably not the best way to learn biochemistry. You just have to decide which one's going to be important. Okay. I've talked about the text. It's available at the Union Bookstore. Has anybody tried to get it there yet? Are they out of it? Not. They were not as of this morning. Okay, so the textbook is in the Union and it is there. The reason I'm sounding surprised is they shipped it on the 12th, so I'm glad it's there. I mentioned the supplementary material we're going to do. Honors designation. In a class like this, a number of the students here are honor students who maybe even find the material not as challenging as you want it to be. I cannot teach this class for the 10% of the class that has had all of organic chemistry and 5 semesters of advanced biology. It wouldn't be fair to the rest of you. So if you want a challenge, or if you want to broaden the course, or if you want to focus on an interest that you've had all along, I warmly encourage you to come and discuss an honors designation with me. If you're an honors student, you can tailor this course to you by any of a large number of possible things, and I'm very supportive of that. Last year we had about 5 or 6 students who did it. One student wrote a short story of a science fiction story. A couple students did papers. One student did a very large poster. You could give lectures. You could do an interpretive dance. There's a wonderful dance of the polyribosome that was done at USC. So there's a lot of different things. I had a student who turned in a quilt for an astronomy class. So I'm really quite open-minded. You may have seen me trying to poke around here. We are, I hope, videotaping this as I talk. All they're seeing is the thing that's on the screen right now. They're not catching all my hand gestures. We will try to have those on the web, hopefully within a day of the lecture. Our intentions are always good. Last year there were a few times when it just didn't work. And they look like they've actually taken into heart the troubles they've had in the past, because they've set it up as an automatic button now. So we're hoping it's more smooth. I mention that because I understand that you may not be able to make a given lecture. A few of you may have lab classes that regularly conflict with one of the 3 lectures, and those videos are principally intended for those purposes. I warmly encourage you to only use them on an occasional basis, that actually coming here where you can ask questions, you can hear other people's questions, is really in the long term better than trying to do it all electronically. The other thing I want to mention is we are a fairly large class. There are clearly enough seats, sort of. We're not going to be able to sit people safely unless people who are early move into the center. I don't know if it's worth trying to fiddle everybody right now, but in the future when you come, go to the center because people need to sit. Academic misconduct is followed under the rules of the College of Liberal Arts and Sciences. I know that almost all of you are here to learn. If it occurs to you to try to take side cuts, I can only say you're not just shortchanging yourself, you're shortchanging your fellow students, you're shortchanging whoever pays for your education, you're shortchanging me from the pleasure of actually seeing somebody learn something, and you're shortchanging the university of a reputation that it has worked hard to try to maintain. If you have complaints, if you have a complaint about any of the people who are helping in the course, please start by trying to resolve it with them, and if that doesn't work, come to me. If you're still unhappy, you can take that to the chair. If you're unhappy with something I'm doing, please come to me, and I hope we can settle it. If not, we can take it up to my chair and so forth. I'm quite open to accommodations, but I'm really, really bad at reading minds. So if you need accommodations of some kind or another, either accommodations during the lecture or accommodations for exams, please let me know, and we will work hard to see to it that anything that needs to be done is done. You've probably all discovered already that these are on a handout here. Is there anybody who doesn't have this handout yet? Want to raise hands? You want to be a good soul and try and deliver these to anybody with a hand up? Thank you. Great. In a general basis, oh, yeah, good. There's going to be one over there, too. On a general basis, we'll have handouts out the first day that we're going to be talking about material, and typically we'll have a few copies the following session as well. There will always be backup copies available at the 4th floor on Bowen Science Building in Biochemistry, and these are also going to be available electronically on the web after we've given the class. Did that just go down? I don't think I meant to do that. Okay. Okay, I guess it didn't go there. Okay. I edited this, and I don't know how this didn't get changed. This is the problem of working with multiple files. Obviously, ignore that specific thing It's Susan O'Donnell who is your true TA. Carissa is definitely available, but not on that schedule. And I will edit the next possible handout to reflect those changes. I am always available after class. I'm available on Tuesday and Thursday at specific times for drop-in. And I'm also available for appointments through my secretary. So please feel free to take advantage of any of those. I'd like to take a moment to find out a little bit more about who you are and what you want from this course, and I'd appreciate as much help as we can get. Want to help out? Two of you can, you two can go to this side from front and back maybe, and Angie and Al to the other side from front and back. Take this to the back and start distributing. What these cards are is a chance for you to tell me a little bit more about yourself. I'd appreciate it if you would put your name, your current class standing, freshman, sophomore, junior, graduate, so forth, your major, your goals afterwards, if you know those, what you want to do after your degree, and what you're hoping for from the course. What is it you want to get out of the course? And anything that you think would be helpful to me in telling you more about biochemistry. Okay, we're still running some short over there. Okay, I'll tell you what, we'll move some up the middle, all right? So you've got some. I'm going to pass these up the middle, and people who haven't got any can. Sure, in fact, I can even write it down. So what I'd like to have are your name, you know, sophomore, junior, senior, etc. Your major, your long-term plans. Now, my reason for this is that if you were all planning to go to graduate school, I would teach this class differently than if you were all going to go to medical school. And if you're some going to do one and some to the other, that's a third way of teaching it. So it does make a difference. And then what you want from the course.
You guys get extras in the middle if you want to pass them out.
Here's some more as well. If you want to help those get around. Great. What you want from the course. Okay. So. Are there any other just general questions about course organization before we move on to actually talking about biochemistry? Yes? Well, we can't post recordings until we've made them. Right, the slides will be posted afterwards. They'll be available in class as paper forms and the electronic version after class. Something else you'll notice is I try to include in the handout any figures I use during the talk because I know how hard it is to try to draw and write at the same time. Many of the figures, in fact, come from your textbook. So if you bring the chapter or two that we're working on, you can have the color version with you there, that way. And the other pictures would be available electronically afterwards. Any other general organization type things before we move on? Okay. I'll let you have a little time for writing because I know people write at different rates, and we'll collect those in a little bit, okay? But let's move on to actual biochemistry. So I've tried to emphasize that what we're going to focus on here is really specifically one broad level of organization, and that is life, but at the molecular level. And you've already heard my quip about being three-quarters biochemistry, but biology coming first. Biochemistry is a deeply satisfying discipline for a number of reasons. For one hand, it really addresses very fundamental questions. How do you transmit information from one generation to the other? How do you adapt to current circumstances? How do you take in energy? How do you, make an atmosphere that people can breathe and so forth? Things that are absolutely bedrock important to life carrying on. There are also obviously many practical applications, and you've probably been seeing these in the news reports at one time or another all along. Advances in medicine, advances in technology, advances in agriculture, many different areas that depend on biochemical insights. Excuse me a second. Sorry. Certainly one important part of biochemistry is what's been called reductionism. The idea that if you know enough about the parts, you can explain the whole. And that has been a very powerful method for a great deal of biochemistry. This little drawing here is showing you how that kind of approach is taken. So if you have some kind of an organism, within that are chunks that perform a suite of functions. Within that there'll be cells, and the cells will be made up of organelles and a variety of molecules. And the hope of biochemistry is that the sum of the organelles and the molecules is going to explain then the behavior of the cell, and then that in turn will explain the behavior of the organ. I will also emphasize, however, there are some things that are better thought of under terms of what's called holism, the idea that the whole might have properties that the individual parts don't have alone. So for instance, a mitochondrion is the smallest thing that can do oxidative phosphorylation. If you breach the membrane, if you take out some of the proteins, you don't get oxidative phosphorylation. So that's the smallest whole that can have that property. The smallest thing that's actually alive is a cell. And so that's one of the reasons that we'll be looking from the cell level down for a great many things. We'll occasionally look above the cell, but not too often. The organization of the text is fairly straightforward. It starts by trying to look at what living things are made out of, the composition of them, and looking at principles of structure, the kinds of chemical behaviors or properties that give rise to structures, and then showing how this core biological principle that structure reflects function is true in this case too. This is one of the underlying truths of all the biological sciences, that structure reflects function. Other sciences are not allowed to talk about function. Astronomers can't say that the function of a neutron star is to emit gamma rays. But we can say that the function of hemoglobin is to carry oxygen. And so function is some behavior or property that is needed for that living system. And almost always the structure of biological things will reflect that function. And so always asking yourself how those two are tied together will illuminate both of them. The next major topic it's going to be talking about is metabolism. And this is, broadly speaking, at either taking in things or energy. How do you go about absorbing and getting into useful form the various things to build stuff with? And secondly, how do you then turn that around and produce the molecules that you need? And then the last major division is looking at what she's called, interestingly, macromolecular metabolism, which I think is a nice shading compared to what it's often called. And here we're going to look at how DNA can preserve or transmit information, sequence information, how RNA can carry it, and how protein synthesis executes the message, and then the proteins themselves do a variety of things. Although there is an extraordinary variety of biological molecules, many tens of millions, if not billions, there's a very limited number of elements that goes into that. And the ones that you're going to see over and over again are hydrogen, phosphorus, carbon, sulfur, nitrogen, and oxygen. Those 6 elements account for probably in the order of 97 to 99% of the mass of living things. And we're going to see why. We're going to see where those atoms fit into the broad classes of biological molecules, and we're also going to see some of the relationships among these. What I've done here is to line them up in the order of electronegativity. So hydrogen and phosphorus are less electronegative. Oxygen is the most electronegative. And electronegativity, you may remember, is the tendency to pull electrons to yourself. So in this sequence here, the further up this end you are, the more you're going to win the fight with the ones down there. So hydrogen will tend to lose some electrons to carbon, carbon to nitrogen, and so on. And that will end up being important when we think about the polarity of structures. It's not an arbitrary property of molecules to be polar. It all comes back to the electronegativity of the atoms it's made out of. There's another question that is worth asking, which is why these 6? And one of the answers is they were around. These are among the 6 most common elements that are available, and so they got used. Carbon, on the other hand, has unique properties, and very likely any life that ever existed or exists uses carbon, if not identically the way we use it, in a very similar way. There are a limited number of functional groups, and I think, let me see, yes. This table is a table of functional groups. That's a lot of functional groups in one figure. You obviously can't read it on the screen. I don't expect you to. This is from your textbook. These are all reviewed. These are all groups you have seen in the past. And so if you're not instantly familiar with what you're seeing in this table, I very warmly and urgently suggest you make up file cards and relearn them. Because this is the letters of the language we're going to be talking for the rest of the semester. These are the little bit pieces that add up into molecules that are interesting biologically. And so having these at your fingertips or on the tip of your tongue will make it possible for you to pay attention to the rest of the things we're talking about. So I'm not going to go over these in any more detail except to remind you that you have seen them all and you will need them through the semester. Any questions about that before I move on? Why don't you pass your 3x5 cards to the sides now? I imagine everybody's had a chance to write. Any other just general course questions at the moment while we have the fluffle and the disturbance? Again, a reminder, when you come on Friday, please move into the center when you come in early. Please leave that patch up there empty for latecomers who really can't avoid coming late. Okay? We're going to be talking about 4 fundamental kinds of biological molecules. There are amino acids, carbohydrates, nucleotides, and lipids. What makes something an amino acid is, surprisingly, having an amine group, specifically a primary amine, and a carboxylic acid group, so an amino acid. This is one representation of an amino acid. This particular amino acid is alanine because of the methyl group over here. And what is important to notice is that in the cell, at the pH that exists in the cell, it will be ionized in this form. So this is the normally occurring form of an amino acid in the cell. And when we get into discussing water and pH, we'll go into that in a little more specificity. But if you see an amino acid drawn without charge on it, that would only be true in the crystalline form. The other thing that makes amino acids what they are is this side chain. Alanine has this little methyl group. Glycine has just a hydrogen. Some things have a whole aromatic ring sticking out there. And we're going to be seeing in a little bit how that side chain confers on them all the properties that are worthwhile to have. I need to get the pointer. Okay. There are a number of ways of drawing amino acids, but this one has been chosen as a compact way of doing it. Some are better at capturing the three-dimensional geometry of them. Others are just good schematic ways of keeping track of the parts without necessarily capturing all of the details of the bonds and so on. Here's an example of alanine in a different representation. This is the ball and stick. This gets a little better at trying to see the three-dimensional character of it. And if you look at that disk in your textbook, you're going to see that you can have it represented this way, and you can now start turning it around so you can see the different parts. The other thing about a ball and stick is that the colors can help you keep track of the elements that are involved. So the gray here is the carbon, and the bright red is the oxygen. Since you need oxygen for fire, that kind of makes sense. And the blue over here is nitrogen, and if you leave your hamburger in the fridge too long, it turns blue. Some way to remember it. And then you've got the little white hydrogens that are not very interesting at all. And if there's any hydrogen advocates here, I'm sorry for insulting your favorite atom. This is actually, in some ways, a more truthful representation. That is, this is a way of showing it that shows the surfaces that would interact with other molecules. Obviously, molecules don't actually have hard, shiny surfaces, but this is the average surface of interaction. So it's a good way of looking at something if you want to know whether things can fit into something else. But if you were to look at this, it would be very hard for you to register the groups involved or the internal structure unless you're very good at it and recognized over here the oxygens and the nitrogens of the carboxylic acid and the nitrogen there. And again, these are all on the CD, and they work really beautifully as interactive ones. This is showing you some of the other kinds of varieties that you can see in amino acids. The side chain chemistry is what's going to make the amino acids so useful. And there are 20 amino acids, and it's because we have a considerable variety, not an infinite variety, a considerable variety of these side chains that we can do with things we do with proteins. So in this case, you have an amide over here. Here you have a sulfhydryl group. So we're starting to get some variety to the chemistry that allows us to do more than a few simple things. The second big group are carbohydrates, and carbohydrates get their name because in the early days of chemistry they looked like hydrated carbon, so that the universal formula for a carbohydrate is CH2O as many times as you need to multiply it. And the common property of carbohydrates is they're polyalcohols. So you can see on this particular representation of glucose 1, 2, 3, 4, 5 alcohol groups on this glucose molecule. So all carbohydrates are polyalcohols. Many carbohydrates can exist in more than one form. So if we look here at glucose, you can see that it can occur in a linear form or in a ring form. And the tendency to form the linear or the ring form will vary from one sugar to another. There's even a further variation here that we'll get into when we get into carbohydrates in more detail. Nucleotides are the chemically most complex of the basic groups. They have all the different atoms basically in them. So you have you have a sugar, a 5-carbon sugar. It'll either be ribose or deoxyribose. You'll have a base of some kind, and this is always a heterocycle that has nitrogen and carbon at least, in addition to having hydrogen and sometimes oxygen. And there'll be a phosphate group, either 1 or 2 or 3. This happens to be a triphosphate. So you can see that a molecule like this incorporates basically almost all of the important atoms. There's just sulfur missing at this point. This particular molecule is adenosine triphosphate, or ATP. It's adenosine because it's an adenine ring on a ribose sugar, and it's triphosphate with a 3 over here. And the authors picked this particular nucleotide because ATP plays so many roles in so much of biochemistry that it's worth getting familiar with ATP and relearning its structure on sort of friendly terms. But all nucleotides will share a base on a sugar with 1 or more phosphates as a nucleotide. If we didn't have the phosphate over there, it'd be a nucleoside without the tide part. And then there's lipids. Lipids are not nearly as easy to describe. They have a lot more variety in some senses of the backbones. They don't have as much variety in the atoms that are in there, but they have a lot more variety in how they're put together. They share the fact that they're not very water soluble. Lipids are sort of generically CH2, more or less. And so here you have a straight chain fatty acid. This is palmitate. It's a fatty acid because there's an acid group up here, carboxylic acid there. That thing can wiggle around like a snake. If you ever saw snakes on the plane, you know the palmitic acids can slide under the seats and everything. Here's cholesterol. No, they would, I mean, if they're big enough. Here's cholesterol, which is also a lipid. Very different kind of structural approach. A little bit of flexibility over here. It's branched, which is a little odd. And then there are these ring structures. So you can imagine that the structural possibilities of cholesterol will be quite different from those for palmitate. But they'll both be poorly soluble in water. And they both resemble hydrocarbons in terms of their overall chemistry. They're CH2 taken as many times as you want to take. Please take a moment. There should be, at the back of your handout for this chapter, a brief diagnostic quiz. This is not part of your grade. This is for you and you alone. It's just meant to give you a chance to touch base about your background in the various courses that I mentioned. These are things that, with the appropriate background courses, you should know. And so take a moment and do it, and we'll see where we stand at that point. You're welcome to talk to your neighbor about it if you want to. Okay. How are folks doing on time? Anybody want a little more time? Okay. Okay. Let's just sort of go over this quickly. For question 1, what will be the correct answer? Great. pH of stomach acid is? Proteins are made of? Okay. DNA is usually found in? Answer would be what? A, B, C, D? Okay. 5, the answer would be A. 6? 7? 8? 9? 10? If there are any of these questions that you've missed, and I'm not going to ask you, you might want to take it to heart and see if there's a pattern to them and be ready to work on getting those out. Okay. Let's move on then. We have a little more time today. I will usually try to start at 3:30. I usually try to stop at 4:20. I understand that you may have to arrive late, you may have to leave early, but I will try to keep to the full time for the other students. So let's try not to get too excited yet. Yes, question? That's an interesting question. She's asking whether I'll have a diagnostic quiz for each section. And it's an interesting idea. I haven't done it in the past. How many people would like to have such a thing? Oh gosh, okay, I'll try to write one then. Okay, I'll do my best. Interesting idea. I always like it when I learn from my students. But in each section, you mean each like block before a test, sort of each several chapters, not each chapter, each section. Okay. Okay. I'll try to see how I would do that. Any other comments, questions, suggestions? Yes. Oh, I'm sorry. The answers. The answers to the diagnostic quiz. The most important difference between prokaryotic and eukaryotic cells is that D, only eukaryotes separate transcription from translation. 2, the pH of stomach acid is 2, A. 3, proteins are made up of a linear sequence of amino acid residues, B. 4, DNA is usually found in the shape of a double helix with nucleotides with the phosphates on the outside, D. Membranes are cells because the lipid bilayer can transport or exclude some molecules, A. Water is a good solvent for B. Polar and hydrophilic compounds. Metabolism is all of the above, D. 8, glucose, fructose, lactose, and sucrose are all sugars. 9, the central dogma of modern biology states that information flows from DNA to DNA and RNA. 10, protein synthesis takes amino acids, energy, tRNA, and ribosomes, letter D. Any other questions, suggestions? Okay. Well, let's continue then with the rest of this chapter. We're here on slide 18 again. And we're actually going to be taking a more leisurely pace of this film, I realize, which is fine. We've talked about the 4 fundamental building blocks. We can only make 3 kinds of polymers. That is, you can make a polymer out of amino acids, you can make a polymer out of sugars, and you can make polymer out of nucleic acids. You don't make polymers of lipids. Lipids are messier than that, as you know if you've done your laundry. Proteins are a polymer of peptides. Polysaccharides are the sugars, nucleic acids, the nucleotides. The common features of polymers is that they have some kind of common shared subunits that are there throughout the polymer, similar without the polymer. They're generally linear, although not always, and we'll see some exceptions to that. Polymers can carry information because they are a sequence that can either vary in the exact composition or it can vary in how it folds up. Polymers have information in them in one way or another. Sometimes not a lot. Glycogen is fairly dull for conversations, but they all carry some information. And then just as a bit of terminology, the residue is the part of the monomer that's still there in the polymer. That in the process of making a polymer, you change the chemistry somehow, and what's left in there is the residue. So it's, strictly speaking, proteins are not made up of amino acids, they're made up of residues. So if we look at polypeptides, and there's a very nice piece in your disc that comes with a textbook that lets you explore this in more detail. They contain, or they're made up of 20 universal amino acids. There are some minor exceptions to this, but there is a universality about those 20 amino acids that far swamps the few exceptions. All organisms can use those 20. Some organisms can use 1 or 2 others. Because polypeptides are a linear polymer of these residues, they can take on a huge variety of shapes, and each particular polypeptide will have a distinct shape, sometimes even more than one distinct shape, depending on circumstances. And they have an immense variability in that structure, from very small peptides to huge peptides, peptides that are 0.5 micron, 3/4 micron long. And what all of this means is that proteins are structurally ideal for functional diversity because of the variety of the chemistry, the variety of the sizes, the variety of the shapes. the kinds of functions that proteins can perform is just mind-boggling. And a good deal of what we'll be looking at, one way or another, is, in fact, that kind of functional diversity of proteins. Polynucleotides, by contrast, are a little bit simpler. They're more kind of focused on the task of conveying information. Any given polynucleotide will have 4 possible bases. So all of them will use A, C, and G. RNA will use U, DNA will use T. Adenine, cytosine, thymine, uracil, guanidine. There are 2 fundamental types of nucleic acid in living systems, the RNA ribonucleic acid and deoxyribonucleic acid. So losing 1 oxygen atom is the huge difference between these 2 things. And we will see in a little bit how much of a difference that makes. Polynucleotides are almost always linear. We used to say they're always linear. Now we know a little more tricks that nature's cooked up. They carry information. And they carry information both by the sequence that can be copied into something else, and also information through their overall shape and form. We didn't really appreciate that so much in the earlier days, but it turns out nucleic acids also have particular structures that are informative, but not nearly as much variety. Proteins have come up with mind-boggling diversity of shapes. Nucleic acids, not so much. Any questions about that before we stop? Because it's 4:20, and I'll see y'all Friday. Good to meet y'all. If anyone needs to come talk about dropping or adding the class, please feel free to come down. And I will be in my office as soon as I've answered any questions here. Just a second. I need to... where is this?